Hi guys! So we've already talked a bit about the cell membrane and its structure, and we're going to talk more specifically today about one of the major functions of the cell membrane, which is transport. So some of the big questions I'd like you to understand by the end of this lecture is: how does a cell control what is transported at the cell membrane? Okay, why is this transport of materials necessary for life? How does the environment influence living systems? And how do cells exist within the confines of the law of thermodynamics? So things spread out. And this is a consequence of the second law of thermodynamics, which states that entropy of a system will increase unless energy is added to that system. So locally, energy is added to living systems, but globally, ever since the Big Bang, no energy is added to the net total of the universe. So we're going to talk about passive transport, which is the movement of material from a high concentration to a low concentration, and therefore there's no energy required. This is the free ride for any molecule. We have diffusion, which is the passive transport of molecules across a semi-permeable membrane. So if we go ahead and have a U-tube, we put a semi-permeable membrane in the center, but it's only permeable to water, not the sugar molecules, these green sugar molecules. We're actually going to see water move across that membrane to balance out the concentration of these solutes. So in the end, after it's sat there for a while, water will have left this concentration to go where the higher concentration of sugars were, so therefore there's going to be an equal concentration on either side. This is called osmosis. It's the diffusion of free water. So you can see this hydrophilic or water-liking sugar molecule has water attached to it because they like each other. You see here the free water is what's going to go back and forth, and it's going to go from an area where there is a lower concentration of sugar to where there is a higher concentration to equal out the concentration of the solute. This can happen when you put dye into water. So if we look up at the top, there's going to be a diffusion of a high concentration of dye to an area of low concentration. So eventually you're going to reach equilibrium. So that's the diffusion of one solute. When you have two solutes, say the orange over here is a higher concentration than this side, and on the right hand side we have a higher concentration of the purple solute. You're actually going to see movement in both directions. So you're going to see the orange move off to the right to try and reach equilibrium, and you're going to see the purple move off to the left to reach equilibrium. So, and what actually happens is that once this reaches equilibrium, they don't just stop. Molecules are always moving. So they're going to constantly move back and forth, but they're going to remain in this state of equilibrium. So chemistry needs to be considered when we're talking about this. So what can actually diffuse through the bilayer, and what does all the other stuff do? So if we took a look, water can actually move through this semi-permeable membrane. Remember, it's hydrophilic on the top, hydrophobic in the center, and hydrophilic again on the end. But water needs to move through at a faster rate than it does through this membrane. And so we have these water channels, which are proteins called aquaporins, which can move mass quantities of water at a faster rate. And that's all along here, as you can see in the cell membrane. This is the same with an ion channel, where it's moving concentrations of ions from a high concentration to a low concentration via these proteins, rather than trying to get through the semi-permeable membrane. Okay, so here we have channels and carrier proteins which control the diffusion. So this channel protein here is pretty much a wide open channel. We've got the hydrophilic protein here. So it's moving from a high area of high concentration to low concentration versus a carrier protein, which actually, oops, say that by mistake, which actually the protein will change its shape to move these solute molecules from an area of high concentration to low concentration. So you see it goes in, it opens up and deposits the solute molecule. Now we're going to talk about active transport, and here you need to pay an energy toll. So The reason why you need to pay the energy toll is because it's movement of material from a low concentration to a high concentration. So it's going against the concentration gradient. And energy is required. You need to use a protein. Why? Well, if we take a look here, we've got an electrogenic pump, okay? And all cells have voltages across their plasma membranes. Voltage is electrical potential energy, a separation of opposite charges. And the cytoplasm of a cell is negative in the charge compared to the extracellular fluid because of an unequal distribution of anions and cations on opposite sides of the membrane. This voltage is called membrane potential. And in this electrogenic pump, proton pumps, the main electrogenic pumps of plants, fungi, and bacteria, are membrane proteins that store energy by generating voltage, so charge separation, across the membranes. Using ATP for power, adenine triphosphate, remember that? Using it for power, a proton pump translocates positive charge in the form of hydrogen ions, here, in the voltage, and hydrogen ion concentration gradient represent a dual energy source that can drive other processes such as uptake. So ATP will come in, it will pay the toll of a phosphate so that the hydrogen ion can be pumped across the concentration gradient. Here we're actually seeing co-transport, which is active transport driven by a concentration gradient. A special carrier protein such as sucrose hydrogen ion co-transporter is able to use the diffusion of hydrogen ions down its electrochemical gradient into the cell to drive the uptake of sucrose. So the hydrogen ion gradient is maintained by an ATP-driven proton pump that concentrates hydrogen ions outside of the cell, thus storing potential energy that can be used for active transport, and in this case we're talking about this sucrose. So it will store up that energy, and ATP is indirectly providing the energy necessary for co-transport. So it uses the energy to pump the protons out. It creates a mass quantity of protons on the outside, which will drive the co-transporter to bring the sucrose into the cell. And ATP is what fills the energy requirement. So phosphorylation, which is the release of that phosphate, triggers a conformational change in the proton. So we can see here we've got a proton pump, the sodium-potassium pump, which is a specific case of active transport. So this transport system pumps ions against steep concentration gradients. The sodium ion concentration, as you can see, which is Na+, is high outside of the cell and low inside of the cell. While potassium, which is the K+, concentration is low on the outside and high on the inside. This pump oscillates between two conformational states in a pumping cycle that translocates 3 sodium ions out of the cell for every 2 potassium ions pumped into the cell. So ATP powers the changes in conformation by phosphorylating the transport protein. That is, by transferring a phosphate group to the protein. So now we're going to talk about tonicity and why it's important. It's a relative measure of solution concentration, and the tonicity of a cell's environment has serious consequences for the cell. Here we go. So if we take an animal cell and put it in a hypotonic solution, meaning that there's a higher concentration of solute inside the cell than outside, water is going to flow into that cell at a high rate, and what's going to happen is that it's actually going to burst. So animal cells don't really like that. Plant cells, on the other hand, do like that because plants love water, and they're lucky that they have this really thick cell wall, which allows a cell to become turgid, which means it's full of water in that vacuole, and it won't burst again because of the cell wall. Animal cells like to be in an isotonic solution, so there's an equal amount of water going in as there is out, this equal amount of concentration of the solute on the outside as it is the inside. So because our cell membrane is so sensitive and at risk of being lysed and broken, it wants to be in an isotonic solution. On the other hand, plants do not like that. It becomes flaccid. If you don't water your plants for a few days, you'll start to see them kind of wilt, and that's due to flaccidity. If we look at a hypertonic solution where there's more concentration of the solute, such as salt on the outside, water is going to rush out of that cell and try and balance out the concentration. And this is when our cells shrivel, and we don't want that. Plants, on the other hand, will get plasmolized, which we definitely don't want our plants to be because there's too much water leaving the cell. Okay, so which way the sucrose is going to go? So if this cell is permeable to sucrose, the cell membrane, then you're going to see it has a 0.03 molarity of the sucrose versus a 0.1 molarity of sucrose. So obviously the concentration is lower on the outside than the inside, so sucrose is going to flow out. But if it cannot flow out of that cell, water is therefore going to rush into the cell to balance out the concentration. Paramecium, which are protists, are pretty cool because they have contractile vacuoles, so vacuoles that can actually contract to pump water in or out. So if it's in a solution where it's all fresh water, water is going to rush into the paramecium. The paramecium does not want to break, obviously. It doesn't want to lyse, it wants to survive. So this vacuole is actually going to contract to pump water out of it. And you'll see this with fish as well. Okay, you'll see a gain of water and salt ions from food and from drinking seawater. The fish can actually excrete salt ions from the gills and salt ions through the kidneys. So there's a water loss through the gills, and it can maintain homeostasis that way. In freshwater fish, you're going to see a gain of some ions in food, the uptake of the ions by the gills, and the osmotic water gain through gills and other parts of the surface. And then there's going to be an excretion of salt ions, a large amount of water, and the dilute urine from kidneys. Okay, so one last thing. Remember, diffusion is simple passive transport through the cell membrane. Remember, these need to be small and hydrophobic. So they're typically small lipids that will go through very easily. There's facilitated diffusion, which does not require energy, but it does require a protein. And as we can see here, this is a good example of an aquaporin, where let's say water is able to go straight through the hydrophilic protein and able to move mass quantities. Here is another example of facilitated diffusion, let's say sugar or glucose, where a carrier protein will actually change shape to move the molecule from an area of high concentration to low concentration. So passive transport, no energy, high concentration to low concentration. Active transport, however, is going against the concentration gradient and therefore requires some form of energy, typically ATP, which pays the phosphate toll in order for anything to be moved from a low concentration to a high concentration. Okay, we've got some bulk transport here that we briefly just want to go over. Endocytosis, which is the intake of large molecules. So this is kind of like phagocytosis, is the cell eating, where it actually the cell membrane will wrap around the food particle and bring it in. We've got cell drinking with this, which is the pinocytosis, which forms vesicles, okay, lining a small, as you can see here, this is a small blood vessel, which is showing that. The receptor-mediated endocytosis, okay, which is just the cell being picky. It needs, this ligand is coming in to the cell, okay, it's got the receptor which says, all right, you can come in, and then it will pinch off, the cell membrane will pinch off around it, and it's coated, and then it's got coat proteins and a coated vesicle. Exocytosis, and you may remember this from going from the rough ER, creating the vesicle to go to the Golgi, where there is modification, and then it exits the cell. So exocytosis, exiting. And it's the release of large cellular products into the outflow. And here's an example of a phagocyte, which I just think is so cool, where it's actually grabbing onto these molecules and engulfing them. And our body does this, our white blood cells do this. It's pretty neat. So if you have any questions, make sure you write them down. Watch this again if you need to, and bring them into class tomorrow. So I just want to make sure you can do these things. Go ahead and take a minute to read it, or you can pause it, and that's it. I hope you guys enjoyed it.